Welcome back. This is Chapter 28A, Part 5. During the process of implantation, the blastocyst begins to be converted into a gastrula. 3 germ layers, as well as an extraembryonic membranes, are developed from the gastrula. The inner cell mass divides into 2 layers, the epiblast and the hypoblast. The subdivided inner cell mass is now called the embryonic disc. Extraembryonic membranes form during the first 2 to 3 weeks of development, and they include the amnion. The amnion is epiblast cells that formed a transparent sac filled with amniotic fluid. This envelops the embryo. It's also called the bag of waters. It provides buoyant environment that protects the embryo. The amnion also helps maintain a constant homeostatic temperature. It allows freedom of movement and prevents parts from fusing together. Initially, the amniotic fluid comes from the maternal blood. Later, fetal urine contributes to its volume. The yolk sac is a sac that hangs from the ventral surface of the embryo. It forms part of the digestive tube, and it's the source of the earliest blood cells and blood vessels. The allantois is a small outpocking of caudal at the caudal end of the yolk sac. It's the structural base for the umbilical cord and becomes part of the urinary bladder, and that's said allantois. The chorion helps form the placenta. It encloses the embryonic body and all other membranes. Here we see a 12-day-old blastocyst. Here is the extraembryonic mesoderm, starting here. Then we have the bilayer of the embryonic disc. We have the hypoblast and the epiblast. This yellow part is the yolk sac. The chorion is beginning to be formed here. The amniotic cavity is this blue part, and the amnion is the dark blue. At 16 days, we have a larger amnion. It's right here. The blue area, the amniotic fluid there. We have the start of the primary germ layers, the ectoderm, the mesoderm, and the endoderm. The yolk sac is larger, and the allantois is extending and starting to form this umbilical cord. 4.5-week-old embryo has a much more developed situation. We have the amnion completely enveloping the growing embryo. It's filled with amniotic fluid. The yolk sac is hanging off here. This is the extraembryonic coelom. The chorion is outside here. And then we still have the chorionic villi. This is the umbilical cord. This is the decidua basalis, and the maternal blood is hanging out in these areas. And then the decidua capsularis. Here's a 13-week-old fetus, much more developed again. We have the decidua basalis on this end. The placenta is shown here in red. The yolk sac is much smaller over to the side. The amnion is dark blue, the amniotic fluid here, and a fully developed umbilical cord. The decidua capsularis is on this end again, and the extraembryonic coelom is there. Gastrulation occurs during week 3, when the embryonic disc transforms into a 3-layer embryo with 3 primary germ layers present, the ectoderm, the mesoderm, and the endoderm. It begins with an appearance of a primitive streak, a raised dorsal groove that establishes the longitudinal axis of the embryo. Cells begin to migrate into this groove. The first cells that enter displace the hypoblast of the yolk sac and form the endoderm. Cells that follow push laterally, forming the mesoderm. The notochord is a rod of mesodermal cells that serves as the first axial support of the embryo. The mesoderm is formed from the aggregation of these mesoderm cells. Cells that remain on the embryo's dorsal surface form the ectoderm, which was formerly that epiblast layer. The ectoderm, mesoderm, and endoderm are primitive tissues from which all body organs are derived. Epithelial cells found in the ectoderm become the nervous system and the skin, epidermis. The endoderm becomes the epithelial linings of the digestive, respiratory, and urogenital systems and associated glands. Mesenchyme cells are found in the mesoderm. The mesoderm becomes everything else. Notice these are all in red and are very important for you to remember. Excuse me, figure 28 shows the formation of the 3 primary germ layers. So here we have this yolk sac right here, as well as the amnion forming here. The cells in between are these cells shown here. So we just have this bilayered embryonic disc. If we take a cross-section of that, we see this. See how they're connected kind of in the middle? And if we take just this section of the 3D view, we can see how they're layered on the outside here with the blue on the inside. Now, this is the bilayered embryonic disc looking down in it. So if we were to take this and then look through it, kind of looking through it this way, this is what we'd see. Okay, this is the primitive streak, which becomes this longitudinal axis of the body. The head end is up here, the tail end down here, and then we have the left and right sections. The yolk sac shown here, and then in the inside we have that amnion. Here we can see at 14 to 15 days how this primitive streak is made. If we took a cut through here, we see the epiblast and then the hypoblast. So they come through here and then out this way. This area where they kind of are fused becomes, at 16 days, the mesoderm. Okay, so the ectoderm comes from that epiblast. The endoderm comes from the hypoblast. And this area between it becomes the mesoderm. Oops, and that's the end.